I would like to begin by acknowledging the Wurundjeri people of the Kulin Nations as the traditional owners of the land on which our Parkville campus stands. I also acknowledge the traditional custodians of the various lands on which you are all joining me from today. I pay my respect to their elders, past, present, and emerging, and to the Aboriginal elders of other communities. Good morning, everyone. My name is Xiaoming, a PhD student from CCEMMP Weihai North. Just a housekeeping note before we begin. Please ensure your microphones are muted during the presentation, and also this seminar is being recorded. Thank you. It's my great pleasure to welcome today's speaker, Dr. Rosemary Cater. Dr. Rosemary Cater is a senior research fellow and group leader at the University of Queensland Institute for Molecular Bioscience in Australia. She fulfilled her PhD under the supervision of Professor Renee Ryan at the University of Sydney, where she used electrophysiology and X-ray protein crystallography to understand how glutamate transporters serve a secondary function as chloride channels. She then went on to complete her postdoctoral training in the lab of Professor Filippo Mancha at Columbia University, where she used the single-particle cryo-electron microscopy and antigen binding technology to determine structures of small membrane proteins. The overarching goal of the Cater Lab is to understand the molecular mechanism of nutrient transport at the blood-brain barrier and how variations to this are implicated in different disease states. Dr. Cater is a current ARC-DCRA Fellow and has received several prestigious awards and fellowships for her research, including the New York Academy of Science Blavatnik Regional Award in 2022 and the Eppendorf and Science Journal Award for Neurobiology in 2024. These achievements recognize her work on one of the most critical yet challenging interface in human biology, the blood-brain barrier. As we know, the human brain is nourished by an extensive network of blood vessels. However, these vessels are linked by the blood-brain barrier, a tightly packed cellular interface that protects the brain, but also pose a significant challenge for nutrient delivery. Today, Dr. Cater will take us on her fascinating journey into how this barrier actually works. She has been structurally and functionally characterizing two critical transporters found at the blood-brain barrier, which are MFSD2A and FLVCR2, which are responsible for importing essential nutrients like DHA and choline into the brain. All yours, Doctor.
Thanks, Xiaoming. I feel like you've given a wonderful introduction. I don't need to give one now.
Thank you.
Oh, no, thank you so much for having me. It's really nice to be able to talk to you all about my research. So as Xiaoming pointed out, I went over to Columbia University and did a postdoc there for 6 years. And then just 2 years ago, I moved to sunny Brisbane to start up my lab at the IMB at UQ. And so what I'm going to be talking about today is the work that I did throughout my postdoc and how that's leading into work that my lab is now continuing on with. So it's all about, if my arrows want to work. Oh no. It's okay, I can do it over here. All right, so we're all about nutrient transporters at the blood-brain barrier. So the human brain is infiltrated by more than 600 kilometers of blood vessels. And like all other blood vessels throughout the human body, these are lined by endothelial cells. So here we've got sort of a cross-section of half of the blood vessel. So we've got the endothelial cell lining it, and you've got the blood up here and the brain parenchyma down here. So endothelial cells that line blood vessels in the brain have a couple of unique features, including reduced rates of transcytosis and tight junctions that hold the proteins very closely together that reduce the passage of molecules from the blood into the brain parenchyma. And it's these properties, along with a couple of others, that are what the blood-brain barrier actually is. So we've all heard about the blood-brain barrier a lot, but I think some people are not can't entirely conceptualize what it means. And so I just like to point this out, that it's actually the properties of the endothelial cells that are the main component of this blood-brain barrier. So the blood-brain barrier, of course, exists to protect our brain from harm. However, it also creates a major bottleneck in neurotherapeutic development because it's really hard to develop drugs that can cross this barrier. But of course, some molecules, such as nutrients, need to get across this barrier to fuel the brain because there's a lot of different nutrients that the brain requires for proper growth and development and aging, but it can't actually make itself. And so in order to get these nutrients into the brain, we have transporter proteins that are specifically matched up to different nutrients. They're expressed on both, it's different for every transporter, but they're expressed on both the luminal and the abluminal sides of the membrane so that these nutrients can be shuttled across the barrier into the brain. And so this is what my research is focused on, is understanding the molecular basis by which these nutrient transporters select different nutrients and allow their uptake into the brain. And also characterizing ones that are not fully understood yet, because we don't always understand how a lot of these nutrients are actually getting in. So we're interested in understanding this not only from a fundamental biology perspective, so I mean just to understand how the brain is getting all of these nutrients that it requires and how these transporters function on a molecular basis, but also because the more we understand about how things like nutrients that can cross the barrier actually achieve that, the more information we have for developing neurotherapeutics that can do the same thing. So there are two different nutrients that my work has focused on mainly so far, which are choline, which is an essential nutrient that maybe not as many people have heard of, but we'll go into that later. But the first one I'm going to focus on are omega-3 fatty acids that I'm sure a lot of you have heard about. So as I said at the beginning, I'm going to talk about what I did in my postdoc, and so we're going to split the talk up into two parts. So the first one will be on the omega-3 fatty acids, the second part will be on the choline, and I'll tell you what I did, and then I'll tell you what the lab is now working towards. So docosahexaenoic acid is one of the three essential omega-3 fatty acids. So it has this chemical structure here. It's got this charged or polar head group and this long hydrophobic polyunsaturated tail. Now, DHA is completely essential for proper neuronal development, functioning, and aging, and it's found in really high concentrations in the brain. So it actually makes up about 5% of the total weight of a neuronal cell membrane, which is pretty crazy for just one type of lipid. However, despite being enriched in the brain so much, the brain can't actually make DHA. And we can make a small amount of it in our liver, but we actually heavily rely on dietary sources such as these, particularly fatty fish, in order to fuel the brain and other parts of our body with this DHA. But of course, as we just went through before, in order to get this DHA into the brain, it needs to cross the blood-brain barrier. And so in order to achieve this, we have DHA transporters that are highly expressed in the vasculature of the brain here. So we can see this here in red. And this transporter is called MFSD2A. So this was characterized by David Silva, who's at Duke NUS in Singapore, and his team in 2014. So it's a relatively...
Do we normally do one meal fraction?
Partake here on...
Fractions. It's up to you.
I did 500 microliter.
Can someone self-partake to please mute his microphone? Yeah, I think he's multitasking in the lab by the sounds of it. But anyway, so as we can see here, MFSD2A is highly expressed in the vasculature of the brain here. So it was only characterized in 2014, so pretty recently. But what David and his team were able to demonstrate is that if you knock out MFSD2A in mice, that it significantly reduces the uptake of DHA into the brain, and this causes severe microcephaly in the brains of the knockout mice compared to the wild type. So you can see they're significantly smaller. So the name MFSD2A it comes from major facilitator superfamily, domain containing 2A. You don't need to remember the name that much. The most important thing is that it's part of this major facilitator superfamily of transporters. So this is one of the biggest transporter families that exist. They exist across all kingdoms of life. And we're really lucky that we have a lot of different structures of MFS transporters that have taught us a lot about the mechanism by which they transport molecules across the cell membrane. So we've got we've got a few example of structures up here and a schematic down here. So these MFS transporters are typically made up of 12 to 14 transmembrane helices, and they're separated into two bundles. So we've got the N-terminal bundle in the more blue colors and the C-terminal bundle in the more red colors. So the transporter starts off in this outward-facing conformation. So we're sitting in this is in the case of an importer. They can also be exporters. They also exist within different organelles, but this is for an importer at the plasma membrane. So these two domains start off in this outward-facing conformation in sort of a clamshell shape that allows substrate and sometimes co-transported ions to come into a central cavity here. Once this happens, we get a conformational change to an occluded state where these two domains become more parallel to each other. And so this is quite a transient state that is visited here, this occluded state, and it exists very briefly before we move to what's called the inward-facing state, where the clam is sort of shifted to this upside-down V shape, where the substrate and co-transported ions, if there are any, can be released to the other side of the membrane. So while we understand a fair bit about how these MFS transporters work, most of the MFS transporters structures solved to date are of ones that transport small and relatively hydrophilic molecules. The substrate of MFSD2A is completely different. So we've got this long hydrophobic tail, and then we've got this zwitterionic head group. And so it's reasonable to consider that the mechanism by which MFSD2A moves a substrate that looks like this across the cell membrane might be somewhat unique, and that it might even involve the cell membrane itself because of this lipidic tail here. So we might actually be using the membrane as some sort of part of the mechanism. So some of you may have just realized that the substrate I just put up there for MFSD2A wasn't just DHA alone. It's actually DHA that's been esterified with a phosphatidylcholine head group. And this esterification is completely essential for transport via MFSD2A into the brain. So if you only have the DHA, it can't be transported. You need to have a head group here. It can be different types of head groups, but it needs to have multiple charges. So it has to be zwitterionic. You can also transport different tails through MFSD2A, but you need to have at least 14 carbons in the length, and some degree of unsaturation is preferred. Also, an important note is that MFSD2A is a sodium-coupled transporter, so it's a sodium symporter. So we need a sodium gradient across the cell membrane that drives the transport process. So our aim here was to understand the structural basis by which MFSD2A mediates the transport of this LPC DHA molecule, or LPC lysolipids they're called more generally, because you'll see further on that we're not actually working with LPC DHA, but we work with other lysolipids as well, but understanding that lysolipid transport. So of course, in order to determine the structure of MFSD2A, we first had to purify the protein. And of course, this is not always straightforward for eukaryotic membrane proteins. And especially when I started this project, it didn't actually start as a Cryo-EM project, it started as a crystallography project. And so we really wanted to get high yields of this protein. So the way we actually went about trying to do that is to try to express different MFSD2A orthologs from all these different species here. So chicken, frog, fish, dog, mouse, human, cows. And as you can see here, we just lyse the cells with detergent after we've done a transient transfection and put the lysate down a small column, and it's got a GFP tag, so we can tell the protein stability and the expression level without actually having to purify the protein. So as you can see here, some of the orthologs had much higher expression than the human one, which is down here in this maroon color. And so we moved forward with the highest expressing one, which was MFSD2A GG, or gallus gallus, so chicken, which has, I think it's 88% identical sequence to that of MFSD2A, and we showed that it transported similarly, and so we moved forward with that for our structural studies. We made a baculovirus for it to amp up the expression and could purify the protein in DDM-CHS quite nicely. And then we wanted to exchange the protein, reconstitute it into nanodisk because of that lipidic substrate. We really wanted to solve the structure of the protein in the presence of a lipid bilayer. And so you can see here that we were able to do that quite nicely. Now, I'm sure a lot of you also know that solving, we didn't last long with crystallography, I should say. So this was in 2018 that this was all happening. And so it was really at that point where Cryo-EM was becoming more commonly used. I wouldn't say it was common yet in 2018, but more commonly used for membrane protein structural biology. But solving the structure of something that's only 58 kilodaltons in size, which is the molecular weight of MFSD2A, that's almost entirely embedded in a lipid bilayer, was almost impossible. We lay a few crystal trays, but then we met Tony Koziakov and his postdoc Satchel here at the University of Chicago, and we came up with an idea to sort of get around this size issue. So what they can do in their lab is they've developed this synthetic phage library, this phage library of synthetically made fabs, by which we can screen for ones that specifically bind to MFSD2A. So we reconstitute the MFSD2A into biotinylated nanodisk and then use phage display to identify fabs from this library that bind specifically to MFSD2A. And so by doing this, we can bulk up the size of our protein to give us better signal to noise, but also we can give us an extra membrane feature to act as a fiducial to help with our particle alignment for Cryo-EM. And so it was really this collaboration that allowed us to move to Cryo-EM for this project. Once I think we got 3 different fabs from Satchel that he sent down to us, and we could see that in our hands that they were binding nicely to MFSD2A. So when you've got MFSD2A in nanodisc, the whole complex is 181 kilodaltons in size by single particle mass spectrometry. When we add the fab, it did size exclusion again. The peak that came out had a molecular weight of 240. 60 kilodaltons difference is pretty close to the 50 kilodaltons that is the size of the FABs. And so we were pretty happy with that and confident that our fab was binding. So then after a lot of optimization on screening microscopes, we were able to obtain some micrographs that had really beautiful particle distribution. If you look closely, you can even see fabs sticking out of the nanodisk, like underneath my laser pointer here is a good example of one. But yeah, we had a large collection of micrographs like that on the Krios, and then from that we were able to obtain these 2D class averages of MFSD2A in nanodisk with the fab bound, just one of the fabs. And so you can see here, this is our nanodisk cloud here. You can see inside there the MFSD2A. You can even see the individual transmembrane helices, which we were really, really excited about, and the FAB sticking out into the soluble space. From these particles, we were able to obtain a 3 angstrom resolution Cryo-EM map. So we can see our N-terminal domain is in the more blue colors. The C-terminal domain is in the more salmony colors, and this is a variable domain from the fab just because we did a local refinement, so the constant domain is not present in the map. And so from that map, we were able to build in our structure, our model of MFSD2A. This was also fun because this was before the days of AlphaFold, so actually had to, look at the map, look at the sequence, figure out, where are these 2 phenylalanines and go from there. And as you can see here, the transporter is in the inward-facing conformation. So we've got this upside-down V shape like this. And what is kind of hard to see in this picture is that there's a large intracellular cavity that's here that harbors some really interesting features that I want to show you. So I want you to imagine that you're standing in the middle of that intracellular cavity and you're looking left, and you can see all the residues here that are being contributed from the N-terminal domain, that N-terminal bundle, and looking right and seeing all of the ones that are contributed from the C-terminal bundle. And so when the first thing that you'll notice is that the N-terminal domain is really charged, whereas the C-terminal domain is quite polar. So it's quite amphipathic, this intracellular cavity, which is quite unusual for an MFS transporter. Looking at the N-terminal domain in a little bit more detail, we saw some interesting features. So first of all, we saw this central charged region, which was sort of this cluster of charged and polar residues that look perfect for binding different types of zwitterionic head groups of our substrate. And it was directly adjacent to the proposed sodium binding site. So this D92 in Gallus, our chicken, is the same as D97 in human. So this is a really beautiful, and not showing all of the data because it's very in-depth, but it's actually a single base cell uptake that uses FACS sorting to measure uptake by MFSD2A that was developed by Choline in David Silver's lab. And this is the proposed sodium binding site because it's conserved to MelB, which this is the sodium binding site in MelB. So these two sites were directly adjacent to each other, which works nicely when you think about it being a sodium coupled binding site. We didn't actually see anything bound there. We were in the presence of sodium, but we're only at 3 angstrom resolution, so that's probably why we can't see any sodium, or maybe it's to do with specific state that we captured. But we could mutate these residues and see that they had some interesting effects. The one I want to point out the most is this E312 residue up here, which can't even tolerate mutagenesis to an aspartate. So it's a very conservative mutation, but it couldn't even tolerate that. Going over to the C-terminal domain, we saw something really cool. So we saw this pocket, quite a deep pocket, that was lined by hydrophobic residues that I've highlighted in pink here. This is just 2 different views that are rotated by 200 degrees. I just wanted you to be able to see the full pocket because it's quite long and twisted. And so when we saw this, we thought, well, hydrophobic residues could form a really nice lysolipid tail binding pocket. And what was even more cool is that we could actually see some sort of non-protein density within this hydrophobic pocket. And so when we saw this, we got really excited because we thought, well, I didn't actually add any lysolipid to my protein sample when I was trying to solve the structure, but we could have pulled down lysolipids or other lipids from the cells that we were expressing the protein in, which in this case was SF9 cells, insect cells. So we went and mutated the residues within this site, and without going into too much detail, we saw a range of different effects, but a lot of the time, if you introduce bulk into residues here that are not normally bulky, then you abrogate transport, showing us that these residues are pretty important for transport. So in order to figure out what this blob was, we reached out to Michael Marty, who's at the University of Arizona, who's an expert in native mass spec for membrane proteins. So he worked previously with Carol Robinson. And what we were able to do with Michael is that if I sent him some of the protein that I was freezing grids of, his grad student James was able to demonstrate that that sample actually contained 2 different species. One that was the APO MFSD2A, the molecular weight is just a bit higher because it's glycosylated, and one that was shifted by 515 Daltons, which was quite unexpected. Now, 515 Daltons is the molecular weight of LPC-18-3. And so we started to think maybe this here is LPC-18-3. We went and looked at the lipidome of SF9 cells, and it is present there. And then we went and did uptake assays to determine if LPC-18-3 is a substrate for MFSD2A, which it is. And so with that information, we built LPC-18-3 into this density. And as you can see here, it fits pretty nicely and matches up well with the stereochemistry. And so just to put that all sort of in context for you here, so we've got the N-terminal domain in yellow and the C-terminal domain in green here. And you can see that the lipid head group is sort of sticking out between these 2 transmembrane helices on the sort of inner half of the protein, as if it's ready to be released into the inner leaflet of the membrane. So we've got TMs 5 and 8, the head group sticks out between those, past these sort of gating residues here, while the tail is poking up into that hydrophobic pocket. And I just wanted to point out that it's actually directly adjacent to that charged central region and the proposed sodium binding site. So when we saw it here, we really thought it looks like it's poised to be released into the inner leaflet of the bilayer. And that was something that we wanted to understand better. And so we reached out to George Kelishvili from Weill Cornell, who's an expert in using molecular dynamics simulations to study lipid transporters. And so George took our MFSD2A, put it in a POPC bilayer, and looked at it under a range of different conditions. And I don't want to go into this in too much detail because I don't have a lot of time, but essentially what I want to point out to you is that he would see that sodium could either go and bind to this E312 residue or the D92 residue here when it was added into solution. And when it went to E312, which we didn't really expect it to do at first, nothing happened with TMs 5 and 8, but when it bound to D92, TMs 5 and 8 moved apart from each other, and this phenylalanine residue would flip out of the way, and that sort of opened up a gateway for the lipids to then move through these two transmembrane helices into and out of the lipid bilayer. So this showed us that the sodium binding at that site is what gates substrate movement into and out of the lipid bilayer. So at that point, we knew a fair bit about what's happening in the inward-facing state. So we knew that where substrate was bound, how it's being released, that sort of stuff. So we wanted to then move on to understanding what the outward-facing state looked like. And fortunately, only about 2 months into trying to look into this, a structure from Liang Feng's group at Stanford came out of the outward-facing structure of MFSD2A. So I didn't actually have to do the work, but we got the exciting results anyway. And so what Liang Feng's group were able to demonstrate is that first of all, they solved the structure of the outward-facing state. They were able to show that so I didn't go into this because I don't really have time, but there's a disulfide cross-link that's present between the N and C domains in MFSD2A that's conserved in MFSD2B, another lipid transporter. But it's kind of weird because these two domains, why would you have them pinned together when you want them to move apart? Anyway, they showed that it's still bound in the outward-facing state. We can talk about that later if you want. But also what they showed is that between TMs 5 and 8, on the outer side of the transporter, they saw a lipid-like density that looked like it was about to enter into the central cavity, which is now an outward-facing central cavity because of the outward-facing structure. And if you remember, TM5 and 8 are the two that we saw lipid leaving between. So it really looks like TM5 and 8 are sort of forming this like gateway into the lipid bilayer to allow substrate to come in from the bilayer and then release back into the inner leaflet of the bilayer. We wanted to understand a little bit more about what that looked like, and so we did some more MD simulations with George, and what we were able to show is that substrate does come in between 5 and 8, also 2 and 11, but more 5 and 8, and then it dives down into the central cavity. You get some degree of substrate of transporter occlusion, but what was really cool is that in that site, that sodium would actually go in and directly coordinate a binding between that E312 residue that sodium really liked to bind to, and we didn't understand why, and the phosphate head group of our lysolipid. So we didn't expect to see this, but also we can now understand why if we mutate this to an aspartate residue, which is a little bit shorter, that you probably can't have this sort of interaction happening. And so that's why that conservative mutation is not even tolerated. And so I'm just going to leave that there for now. So essentially between our work, our collaborators' work, and other people in the field as well, we've been able to really tease apart the mechanism of transport for LPCs via MFSD2A. So we know that it comes in from the outer leaflet between TMs 5 and 8, dives into the central cavity, is flipped during that process, and then released between TMs 5 and 8 into the inner leaflet. So this was a big collaborative project that I thanked a lot of people throughout the talk and acknowledged them for their work throughout the talk, but a couple of people that I didn't acknowledge throughout the talk were Felipe Mancha, who was my postdoc advisor, who gave me everything that I needed and set up the collaboration with David for this beautiful piece of work. Brendan Choi, who was an undergrad at Columbia, who worked with me on this project for 4 years, and Ollie Clark, who, aside from being a fellow Australian in New York, was also, he taught me everything I know about Cryo-EM data processing. And so the question is, what's next then for MFSD2A? To part 2, where we're going to be talking about choline. So after I'd done all this work on MFSD2A, I was really interested in, these nutrient transporters of the blood-brain barrier and trying to figure out, what am I going to work on next? Because I don't have to solve the outward-facing structure of MFSD2A anymore, so what am I going to do with my time? So I was on Twitter one day, which was still a thing, and I think this was 2022. And I came across this paper that said lack of FLVCR2 impairs brain angiogenesis without affecting the blood-brain barrier. So you can see here, if you knock out this gene, FLVCR2, then you don't get blood vessels forming in the brain here, whereas in wild type you do. And I thought this was really interesting because I knew that FLVCR2 was an MFS transporter. There isn't a whole heap else known about it though. And so we know that mutations in this transporter cause what's called Fowler syndrome, which is a very rare disease, but it's characterized by cerebrovascular malformations and hydrocephalus, so buildup of cerebral spinal fluid on the brain, but no one really knew what it transported. There was some ideas out there that it might be a heme transporter, but it hadn't been directly demonstrated. And so it was really still a question that was up in the air as to, what is this FLVCR2 protein transporting and how is it having this effect? And so after seeing this paper, I popped down to Filippo's office and I said, hey, Filippo, do you mind if I reach out to this Tom Arnold guy at UCSF to see if he wants to work with me on this? And he said, yeah, sure. Go for it. And then I did. And then about 10 minutes later, Tom, who is just one of the best collaborators I've ever had, replied saying, yep, I'm keen, let's do it. And so we went down this whole pathway, of trying to figure out what the substrate actually was. So we're going to set up all these metabolomic screens and whatnot, but then at the meantime, trying to express and purify the protein. But about a year into this, these two papers came out in Cell Metabolism back to back, demonstrating that FLVCR1, which is clearly quite related to FLVCR2, don't worry about the name. It's actually because it's a viral receptor that it has this name, but it's not a very good name. So anyway, the subtype 1 was demonstrated to be a choline transporter. Now, FLVCR2 and FLVCR1 are 55% identical, but they have different expression patterns. And so at this point, we're like, well, maybe FLVCR2 is just a choline transporter as well. Maybe we don't need to do all this metabolomic screening. And so just to take a step back, because we're now talking about a couple of new things, this FLVCR2 and choline. So let's just talk about choline for a minute. So what exactly is choline? So as I said at the beginning of the talk, it is an essential nutrient, but it was only determined to be in 1998. And there's still a lot of active research being done on exactly what choline is doing and when it's really important. So it has this quaternary ammonium group and then a couple of carbons and a hydroxyl, and we need to get it from foods such as these. Eggs are particularly rich in choline. And so we need to consume about 0.5 gram of it a day. And if you're pregnant, you need to consume about 700 to 800 milligrams of it a day because you're trying to grow another human. And the reason we need so much of choline to grow another human is because choline is essential for making phosphatidylcholine, which is the most abundant lipid in all of our cell membranes. So you can think about just how essential it is that we get choline in terms of, cells proliferating in order to make phosphatidylcholine to support cell growth. We also use choline to make betaine, which then gets converted to something else called SAM, which is a universal methyl donor. So it's really important for epigenetics in terms of DNA, protein, RNA methylation. And also we need it to make acetylcholine, which is a really important neurotransmitter. And so choline is very important. It's particularly important for the central nervous system, as I said, particularly during embryonic development. But how choline actually gets into the brain is very poorly understood. So the only two bona fide choline transporters that we have so far in our knowledge base are FLVCR1, but FLVCR1 is not expressed in brain endothelial cells. It's more peripherally expressed and expressed in neuroprogenitor cells. And the CHT, which is a high affinity choline transporter, which is almost exclusively expressed in cholinergic neurons. But FLVCR2, at least on an RNA level, is expressed in brain endothelial cells. And so this started up our question of, is FLVCR2 a blood-brain barrier choline transporter? And so together with Tom's lab, we used mouse models, cell-based assays, liposome assays, and structural biology to try to answer this question. So starting off by the work that was done by Divyanti Mukherjee, who's a really talented postdoc in Tom Arnold's lab at UCSF. So she developed a couple of really important mouse models that were really critical for our understanding of what FLVCR2 is doing. So the first one that she made was a mouse that had FLVCR2 that was had a hemagglutinin tag attached to it, so that we could image on a protein level where this was being expressed. And as you can see here, it was being very highly enriched in the microvasculature of the brain, and it was actually expressed on both sides of the cell membrane. So if you have your endothelial cell, the side that faces the blood, so the luminal side, and the abluminal side, the side that faces the brain. So in a great place if you want to take up choline into the brain. She also developed a conditional knockout of FLVCR2 so that she could let the mice develop their blood vessels properly and then knock it out in adulthood and inject radio-labeled choline into those mice and controlled mice to see what would happen. When she did this, she saw that the knockout mice had a significant reduction in the uptake of choline into the brain, which was really exciting because it was our first bit of data that demonstrated that this was a choline transporter at the blood-brain barrier. She also did a range of different cell-based assays that further confirmed our hypothesis that FLVCR2 is a choline transporter. I'm not going to go into them all in detail, but essentially from this and also some liposome-based studies that we did, we were able to demonstrate that FLVCR2 is a choline transporter that regulates choline uptake into the brain. So we then wanted to understand how this looks. What does this look like? How is it binding choline? How is it transporting choline? And so we went through the same process of expressing different orthologs of FLVCR2. Here, the Bos taurus, the cow, and the Mus musculus, the mouse, both expressed quite nicely, but we went forward with using the mouse given that all of Dibianti's studies were being done in mice as well. We were able to purify, we amplified the expression using a baculovirus system, and then we're able to purify the protein in TDM-CHS. And we could also reconstitute it into nanodisk, which we did in order to do the exact same trick that we did with MFSD2A, again with Tony and Satchel, to try to bulk up the size of our protein. In this case, we actually got 40 different Fabs identified that seemed to bind to FLVCR2. Obviously, we didn't ask them to make all 40 and send them to us, so they just sent a few. They sent a couple that had quite a high affinity for FLVCR2. And so we asked them to send us those. Again, we showed a nice shift in this place. It was actually closer to 50 when we added our FAB, and we could see shifts in our size exclusion chromatography profiles to show that the Fab was binding in our hands. Now, while we did this in nanodisk, when it came to actually determining the structure, I wasn't able to do this in nanodisk because the protein was highly flexible in nanodisk. So when I did this, the protein, I could pull out different conformations from the one data set, so an outward-facing state, an inward-facing state, and a partially occluded state, but these were all quite low resolution. And this low resolution was not only impacted by the flexibility, but also because I had a really severe preferred orientation issue. And so after trying a range of different things, I actually just ended up, going the easy route and just keeping the protein in detergent, which seemed to essentially solve the problem. Aside from in detergent, the ice gradient is really tricky to work with. So you might have one hole with really beautiful particles, the one next to it has none. So aside from having to sort of brute force it a little bit, it solved our problem. So we took our FLVCR2 in detergent, added our Fab, added some choline so we could understand how it's binding, optimized our grids, and then had a Krios collection where we were able to obtain, as I said here, the ice distribution. This was probably the best out of all, and they didn't all look like this, I'll show you that much. But we got some nice particles, mostly side views, but side views from different sides. So we're still able to get a nice quality map from this. And so here we were able to use these to get a 2.5 angstrom resolution map of FLVCR2. And here the transporter has been captured in the outward facing state. So you can see it's got that outward facing V shape here, or clam shape here. And then this is the FAB up here in the gray. We're able to build our model into this. And when we did this, what was really cool is that we could see a density in the central cavity that we thought might be choline. However, I say might be choline because it wasn't the most beautiful density ever. There was definitely something there, like the hydroxyl in particular, the density for the hydroxyl was really strong and was very closely connected to that asparagine residue, but I expected it to be nicer for a 2.5 angstrom resolution map. And so despite being very excited that we had a structure of FLVCR2 with probably choline bound, I was a little dissatisfied with this and couldn't quite understand at first why this was the case. But then if we go back and look at where that FAB is bound, it's bound directly between TMs 2 and 11, which are mislabeled here. That's 11 and this is 2. But my point is that it's sticking right in between those two transmembrane helices. And if we go back to these rocker switch, this is from the C term, this is from the N term, and they need to come close together in order for the transporter to occlude. And so what's probably happening here is this Fab is acting as an allosteric inhibitor of the transporter, and so preventing the transporter from entering, even in an outward facing state, what a proper binding site would look like, let alone an occluded. And so with that, we were like, well, maybe if we have a different Fab that binds to a different site, we'll be able to not have this inhibition happening and be able to obtain a better resolution density for our choline. Unfortunately, one of those 40 did. So there's just an epitope binning assay which demonstrated that this Fab9 bound to a different site. Fab9 had a pretty rubbish affinity for FLVCR2, but when we put it on size exclusion, like when we complex it with FLVCR2, our peak, which is the yellow one here, compared to without Fab, was still shifted to the left, indicating that it was, you know, the particle size was increasing. And so we decided to give it a shot anyway. And so we went through the same process, but with the different Fab. And from that, we were able to obtain a second Cryo-EM structure of FLVCR2, which we can see here. And in this case, well, first of all, the Fab did bind, so that was exciting. But in this case, the transporter was in the inward-facing conformation, which was not expected, but pretty exciting that we were able to get high-resolution structures of both the outward and inward-facing states. When we built our model for this, we got a much nicer density for our choline in the central cavity there. And as you can see here, we've got this sort of aromatic cage that's forming around the choline. So we've got this tryptophan residue, and there's been mutations done on these. They're all pretty important for substrate binding and transport. But yeah, we've got this aromatic cage forming here, and then these hydrogen bonding networks that are happening at the back with the hydroxyl. So this is really cool to see. But what's also really nice is that this aligned really well with what else has been seen for choline transporters. So we published this work back to back with the structure of FLVCR1 from Rich Height's lab, which showed it essentially in exactly the same site. The only choline structure saw before was this LIC-B, which is the bacterial choline transporter, which also has an aromatic cage. During the revisions for our paper, this high affinity choline transporter structure got solved, which also has this aromatic cage, which is really nice. And so with that, I'm going to leave that part for the FLVCR2 and just say that we were able to demonstrate that this is a choline transporter at the blood-brain barrier and reveal molecular insights as to how this actually happens. First of all, thank you all for your time, but also just to say that towards the second half of this year that I'm going to be recruiting a postdoc and/or a research assistant. So if anyone likes the sound of the sort of research that I've been yabbering on about for a long time now, then please send me an email, because we'd love to have you come join our team, which I'm really proud of the team here. They're starting to do some really excellent work. And yeah, we'd love to have you come join if it's something that you're interested in. And so with that, I will finish, and thank you for your time.